Okay, in this session I'm going to be talking to you about 3D cell culture models, and in this session I will take you through some recent research that we've done in our lab using various different 3D cell culture models to show you the scope of experiments that can be performed with those models. So this is what I'm going to cover in this session. Why do 3D cell culture? We're really wanting to mimic normal cellular environments, which will have both extracellular matrix and cell-cell communications, and also very importantly hypoxia. So I'm going to talk a fair bit about hypoxia today. I'm also going to cover the various different methods that are available to do different forms of 3D cell culture, and I'll show you some of the recent models that we've developed in our lab to do very specific experiments on cells in 3D cell culture. So the main reason that we do 3D cell culture is because we know that in cells grown in standard 2D cell culture, so that's cells grown on a plastic surface with a liquid overlay of growth medium, this doesn't reflect the 3D nature of tissues where you've got cell-cell interactions, cell-extracellular matrix interactions, and hypoxia to some extent, and more limited oxygen and carbon dioxide levels. So in 2D cell culture, when they're just grown on a plastic surface, if you're growing cells in that way and you treat them with some drugs, for example, every single cell has completely equal amounts of oxygen, fetal calf serum, nutrients, loss of waste products, and the cells also don't organize into their normal 3D arrangements. So this is a very artificial way of studying cells. When we grow cells in 3D cell culture, we tend to grow them in some sort of sphere-like shape, and growing them as a sphere allows you to set up 4 different environments which mimic the normal environment of tissues, or certainly some of the normal environments of tissues. Now, if you allow these spheres to get big enough, you'll develop a hypoxic core in the middle, so this is where cells are completely devoid of enough oxygen to survive, so you tend to get either apoptosis or necrotic cells in there. We then have a chronically hypoxic layer. It's labelled here as PMO positive because PMO is a stain that we use. Pimidazole is a stain that it changes its chemical structure in the presence of oxygen, and then we can use an antibody to detect regions of tissue which are hypoxic. So this would be a PMO positive or hypoxia positive region. We've then got typically a low proliferation, reasonable levels of oxygen ring around that, and then on the very outside edge we tend to have very high levels of proliferation, high levels of oxygen, and these cells are basically growing very, very fast because they have got nothing to limit their growth. So you can set up all of these 4 different levels within a 3D cell culture, and therefore you can study how cells behave at these different levels. Now, one of the main things that limits cell growth is presence or absence of oxygen, and also to a similar extent glucose. So concentrating on oxygen, we know that and we covered this in the stem cells lecture, when cells are low in oxygen, they can secrete vascular endothelial growth factor, and that will encourage new blood vessels to enter a region of hypoxic tissue. And this whole process is controlled by hypoxia inducible factor, which is a transcription factor which is either present or absent depending on the level, on the partial pressure of oxygen within a cell. So HIF, hypoxia inducible factor, it's this transcription factor that binds to the promoter of genes that are required when oxygen is low, so vascular endothelial growth factor, for example, also GLUT1, which is a glucose transporter. So it induces transcription, hence it's called a transcription factor. And it is constantly produced, so your cells are always making HIF. However, to allow a oxygen-dependent transcription of gene expression, HIF is degraded in the presence of oxygen. So, if oxygen's high, HIF is targeted for degradation, and this is at the proteasome. If oxygen levels are low, this HIF degradation by the proteasome is blocked, and therefore we get HIF responsive genes. And this is just shown here. So here we've got, in the presence of hypoxia, we've got HIF1 alpha, which is constitutively expressed, and this forms a dimer with HIF1 beta also called ARNT. But this will switch on gene expression such as vascular endothelial growth factor and glucose transporter GLUT1. So that's in the presence of low levels of oxygen. In normoxia, normal levels of oxygen, we get the presence of oxygen allows HIF1 alpha to have these OH groups added. So this is prolyl hydroxylation. There's an enzyme, prolyl hydroxylase, which adds these OH groups. That means that HIF1 alpha is now recognized by VHL, von Hippel-Lindau protein, and that acts as a E3 ubiquitin ligase to encourage HIF1 alpha to be degraded by the proteasome. So in normoxic conditions, HIF1 alpha is degraded and the cells know that they have got oxygen. In the absence of oxygen, this can't happen, and then the cells will switch on hypoxia response genes such as vascular endothelial growth factor. And this is just summarised here. VEGF, we know that VEGF stimulates new blood vessels to move into hypoxic areas. GLUT1 is also a glucose transporter. Wherever you've got low levels of oxygen, you've most likely got low levels of glucose as well. And this is important for some later slides where I'm going to show you some GLUT1 staining across some of our 3D cell culture constructs. So hopefully now you've got a sound idea of what's going on within these 3D cell cultures. On the outer edge, the cells are quite happy. On this ring of cells here, just under the outer edge, the cells are getting on, they're dividing. They've not got unlimited amounts of glucose and oxygen. They're maybe starting to get a little bit stressed. In this region here, they are chronically hypoxic, so they're expressing vascular endothelial growth factor and they're upregulating their GLUT1 transporters. And these cells in the centre have got so little oxygen and glucose that they are most likely dead. So what I'm going to go through now are several different methods to generate 3D cell cultures, to create cells in like a sphere-like ball of cells that will give us that hypoxic centre and the normoxic outer rim. One of the easiest methods to use is called the hanging drop technique, and as the name suggests, we have a drop of liquid hanging from something containing cells. Now cells like to stick to plastic, and when we grow cells in normal cell culture, they stick to the plastic aided by the extracellular matrix proteins that are present in fetal calf serum. So when we grow cells in normal cell culture, we put in fetal calf serum that contains growth factors, but it also contains extracellular matrix proteins, and these allow cells, that sticks to the plastic and it encourages cells to stick to the plastic. If you grow your cells in a ball of liquid medium hanging from a surface, and this is the easiest way, it's just a petri dish lid, you put little 10-20 microliter balls of liquid medium containing cells onto the lid of a petri dish, and then you quickly flip it over and you get surface tension, and these little balls of liquid medium stay as little balls of liquid medium and the cells are within there. Now they have got nothing to stick to. They've got no plastic to stick to. They'll fall to the bottom of this dome of liquid and they will stick to each other. So that is a tumour, or a cellular spheroid there within that ball of liquid media. So this is the quick, easy way of doing it with a petri dish. You can also buy inserts that fit into 96 well plates where you pipette your liquid. You've got these little, this is a cross section through an insert that goes into the top of a 96 well plate, and the liquid just hangs in this little cavity here and we get a spheroid forming. What we can then do once these spheroids are formed after about 5 days is tap the plate and this drop of liquid drops into the 96 well plate down below, and then you can do your experiments. Similar idea to that is the use of low adherence plates or low adherence substrates. So as I've just told you that cells like to stick to plastic, what you can do is buy plastic plates which are coated in such a way that cells can't stick to them. And there are two ways of doing this. One, buy the very expensive plates which are almost coated with a polymer which prevents the cells from sticking, so the cells have got no option but to stick to each other. Or the cheap method is you can coat the bottom of a 96 well plate with a bit of agarose, and the cells really don't like sticking to agarose. Either way, you end up with a sphere of cells. You can buy these little special inserts. that are microscopic, so these will take about 20 microliters of cells and create thousands and thousands of tumour spheroids. So these are sort of commercial 3D cell culture plasticware, very expensive, but they work very, very well. Now, once you've got your cells growing in 3D cell culture, you want to do some sort of analysis on them. And the sorts of things that we routinely do is section through the 3D cell cultures as if they're a piece of tissue and then stain them up by immunocytochemistry. So this is just some irradiated cell cultures. They've been, so this is controlled, 2 grays of radiation, and in that case 4 grays of radiation. This is 2, 4 and 6 grays of radiation. Grays is just a unit of absorbed dose of radiation. So in this experiment we've got cyclin B1, and you can see cyclin B1's predominantly expressed around the periphery where there's high levels of oxygen, high levels of glucose, and less so in the middle. And you can see in the middle it's quite cell sparse because this is the hypoxic core. This is looking at an apoptosis marker, so activated caspase 3. And what you can see here, there is some caspase 3 here, and these are at the bottom, these are some radiosensitive spheroids. So you've got masses of caspase 3 here, less so here. So there is still some caspase 3 because these cells are chronically hypoxic and very, at this point, very unwell. But in this situation, we've irradiated with 6 grays of radiation, and you've got a massive upregulation of caspase 3. So you can do analyses on 3D cell cultures much in the same way as you can do the same sort of analyses on cells grown in 2D cell culture. You just have to section them first. So the hanging drop and the low adherence plates that you've just seen, so these and these are some of the most commonly used assays for 3D cell culture. In our lab we tend to focus on the Alginate bead assay, which is an alternate one. This is what we call a clonal growth assay. Both of these two are aggregation assays in that you can put 10,000 wells in here and just basically stick them together into a spheroid. And the same true with the low adhesion plates. Whereas the Alginate bead assay is what we call a clonal assay, where each individual colony is formed by a single cell with its own proliferative ability. So these aggregation assays, you don't necessarily need proliferation to form a 3D cell culture, whereas in the Alginate bead assay, you do. So alginate, it's a polymer extracted from seaweed, and it's really useful in 3D cell culture studies because it acts as a scaffold, and the reason it's so useful is it can be cross-linked by calcium. So this just shows the charge groups on alginate. It's just a polymer of carbohydrate, but it has got lots of these negative charges, and it's in big long strands, and if you add in a calcium 2+ ion, it will cross-link one strand to another and reversibly cause this solution of alginate to form into a gel, a clear gel. So what we do is we mix our cells with a solution of sodium alginate. We then drop those cells into a solution of calcium chloride. That will cross-link the alginate, and then individual cells will be encased within alginate, and then we will put that alginate bead into some liquid medium with usual growth factors. So a typical alginate bead is about 5 mm across. It will be impregnated with thousands of cells, and then we allow those to grow, and every one of those cells which has proliferative ability will then form a microtumour spheroid. Now in the learning materials folder I've put a YouTube video on there of me making some alginate beads, so you can see the whole process, which involves the first stages of cell culture all the way through to creating alginate beads and 3D cell cultures. And this is what they look like down the microscope. These are being stained with Hirsch 33342, so you should all know that Hirsch 33342 stains nuclear material blue, and it can get into live cells. And it's also been stained with prapidium iodide, which is a vital dye which can only get into dead cells, and it stains them red. So on day 1, after setting up an alginate bead, you can see that most of the cells are just single cells, as you'd expect. By day 4, you tend to get clusters. I think this is day 2 there. Day 7, you've got larger structures up to 100 microns in diameter, and then by day 14 you've got these great big structures of a couple of hundred microns in diameter, and if you leave them to go for 3 weeks, you tend to get these structures plateauing at about 500 micrometers across, so that's half a millimeter. The center of that will be containing both live and dead cells. The periphery will be proliferative and relatively happy. And what we do is when we look at the size of these, we tend to get a plateauing at half a millimeter. That's as big as you can get these things to go, which for most applications is okay. For some applications is not quite big enough. This is just one of our recent studies using the 3D Alginate assay, in this case to assess the effects of the pro-apoptotic agent Trail with a histone deacetylase inhibitor. I'm just showing this because it shows that we can take look at cross sections through these individual spheres and assess their nuclear morphology for apoptosis. And what we found here is that in 3D cell culture we got a potentiated response to the drugs that we were interested in. You'll see a little bit more of this later. Now these constructs here are about 100 micrometers, maybe 200 micrometers across. These that I've just shown you previously are up to 500. For assessing apoptosis, that's more than enough. But for some applications that we do, we need much bigger constructs. We have a whole program of research that involves mass spectrometry imaging. And mass spectrometry imaging involves looking for metabolites and proteins across a tissue construct. And we are limited by the diameter of the laser that we use to excite molecules. So basically, if we perform mass spectrometry imaging across a small construct like this, and then looked at what metabolites were present, we would literally get about 3 or 4 pixels of data across each of these structures. So what we've done is invented a much larger structure, which is up to a couple of millimeters across, that allows us to create images of what metabolites are present in 3D cell cultures to mimic human tissues. So to do this, we've created a new cell culture model that we've termed Aggregoids. Okay, so this Aggregoid here is stained with Hirsch 33342 as before, and propidium iodide, and you can focus on the surface, and you can see that there's, proliferative cells, and there's a hint of red in the background, and so we know we've got a hypoxic core. So the way that we make these is we use a combination of the alginate bead assay and an aggregation step. So what we do is we grow lots of cells within an alginate bead, and within that alginate bead we have lots of tumour spheres growing. Because the alginate is cross-linked with calcium, we can add in EDTA that will chelate the calcium and dissolve this alginate gel to then release hundreds of tumour spheres, each at about 100 micrometers across. And then what we do is we aggregate those together into a large spheroid aggregate. So we start off with hundreds and hundreds of small spheres. We put them into an agarose coated well, and we've seen agarose coated wells. They were in this low adhesion plate experiment here. So we create a low adhesion plate, and then instead of adding in hundreds of cells, we add in hundreds of spheres, and then we get a much larger aggregate forming. And this is what they look like. They're a couple of millimeters across, they have a proliferative rim, and they have regions of apoptosis where the cells are really quite stressed, and they're also positive for caspase 3 activity because we've got apoptosis going on in the center of that construct. So these are some analyses done on our Aggregoid model where we've spent a fair bit of time characterizing the model and seeing how it works. This is a lung aggregoid that has been stained up simultaneously for 4 different proteins. And the technology behind doing multiplex immunos here, we've actually done mass spectrometry imaging to image multiple proteins simultaneously, but I'll not burden you with that too much. But what we can see here is we've got green, which is ecadherin. Importantly, red, we've got GLUT1, which is a surrogate marker for hypoxia, and you can see that this is hypoxic in the center. We've got purple, which is your Ki67 proliferation marker, generally around the edge. and in blue we've got DNA damage, lots of DNA damage in the centre, and that is gamma H2AX. It's a histone modification. Similar with the osteosarcoma model, we've got, in this case, Chi67 in green around the outside edge, and GLUT1 tends to be more in the centre. We're really interested in these two models of osteosarcoma because one of them shows a hypoxic centre. This other model shows hypoxia all the way across the aggregoid, even on the edges. And what we think is going on with this model is that it's got a defect in its hypoxia response, which is helping to drive it to grow abnormally. So we're currently studying these two models in more detail. This is just that lung picture in more detail, and we have recently just published this paper in Analytical Science in 2020 showing the full characterization of the protein expression of in these aggregoids. And in that same paper we've also looked at metabolites. Now I've talked about hypoxia quite a lot early on, and the reason hypoxia is really important is because where there is oxygen you can have oxidative phosphorylation. When there isn't any oxygen you have to do glycolysis. And what we have shown in this paper using these aggregoids is we've looked for metabolites by performing mass spectrometry imaging and looked for different metabolites across these aggregoids. And what we can see is wherever you've got metabolites involved in the TCA cycle, so citrate, succinate, malate, you have got the presence of those metabolites only in the well-oxygenated rim, whereas when you have metabolites associated with glycolysis, so you've got lactate, pyruvate, and also these here, particularly this glutamine pathway, is particularly relevant in the absence of, particularly lactate is a classic marker for the absence of oxygen. When you metabolize glucose in the absence of oxygen, you tend to generate lots of lactate rather than going into the TCA cycle. So what we've shown using this model is the metabolites that can be visualized in our 3D cell culture model make perfect sense when we look at the TCA cycle. So we're very confident that we've created something that allows a study of different phenotypes within tissues. Now, everything I've been talking about so far has been really relevant to the study of cancer in cell culture. There are some models of 3D cell culture which are very much designed for studying normal cells and in particular stem cells. And one of those is organoids. So organoids are where you take tissue, typically take tissue and isolate tissue stem cells. So we learned about stem cells in recent weeks. And then you isolate these stem cells and grow them in a gel. It's called Matrigel, but it's an extracellular matrix gel that supports stem cell-like behavior. So that gel contains the appropriate extracellular matrix proteins for maintaining cells in a stem cell-like phenotype. And we heard about the stem cell niche a few weeks ago. And what you can do is by limiting the growth, certain growth factors, so limiting, certainly limiting things like serum albumin from fetal calf serum, but putting in growth factors that promote stem cell behavior, you can promote stem cells to remain stem cell-like and then create micro tissues within a cell culture environment. And we call those organoids. They're called organoids because they're organ-like. You can take a single stem cell and differentiate it down multiple different lineages to create something that resembles the normal tissue for which that stem cell came from. And this is an organoid of human cortical brain. And in this situation, they've used induced pluripotent stem cells. That's taking differentiated cells and de-differentiating them back to being stem cells by making them express these stem cell transcription factors. And then you can grow these cells in a Matrigel extracellular matrix gel type substrate, limit the growth factors that it receives, put in stem cell promoting growth factors, and a single cell can turn into multiple different cell types, all of which are normal-like, and in this case create a model of the human cortical brain. And in white are neuronal cells forming neuronal connections with other neuronal cells, and within there you've got all of your other support cells such as oligodendrocytes, astrocytes, and so on. So all of this can come from a single cell that has the ability to differentiate down multiple different lineages. So organoids are really sort of fascinating models of any human tissues, and you'll see organoids that have been developed for breast tissue, prostate tissue, most of your epithelial tissues, but also these brain organoids. These are really sort of almost like studying the very early developmental steps of brain development. So these are fascinating things, and if you ever get the chance to work on organoids, take that opportunity. And the final model I'm going to talk about are models of human skin, and this is more work that's been done in our lab. We work on creating full thickness skin models for non-animal testing of drugs and cosmetics. So this full thickness skin model that we create, we mix fibroblasts, which are the underlying connective tissue producing cells, with keratinocytes, which are your surface skin cells. So your very surface skin cells are epithelial cells, they're called keratinocytes. Then there's then a basement membrane, and then below that there are fibroblasts, and this is where you would normally find your blood vessels and nerves as well. So it's a two-layer system. So this is normal skin. This is the cornified, keratin-containing, dead, dried out keratinocytes. These are the proliferating cells. The basement membrane is about here, and then these are your fibroblasts. This is what our artificial skin model looks like in cross-section. It looks superficially very similar. We've got this cornified layer here, and this is what it looks like in how we grow it. It's grown in 6-well cell culture plates, and these are what we call a millipore insert. So basically it's a 6-well plate, there's liquid medium in the bottom, there's a plastic insert that sits in that liquid medium that just touches the liquid medium, and there are tiny little holes to allow liquid medium to go from the bottom through the membrane and into this top chamber here, which is where the cells are grown. So it's called an air-liquid interface model because the skin is an air-liquid interface tissue. It's fed by liquid from underneath. So what we can now do is, by isolating keratinocytes and fibroblasts, usually from donated skin, from excess skin from surgery, patients will allow that excess skin to be used by a tissue bank who can isolate the cells and then post them down to us. We can then create an artificial skin model, and then we can perform experiments and set up models of human disease. So we're currently working on models of melanoma, models of psoriasis, and models of dermatitis. And we've recently published this study on using this technology to create a 3D tissue engineered model of psoriasis skin, and we do that by stimulating the cells within this structure with cytokines that we know induce keratinocyte proliferation. So we particularly put in IL-22 that induces epidermal keratinocytes to change into psoriasis, and we get psoriasis-like plaques forming, and then we can do experiments on psoriatic skin versus normal skin in a cell culture setting without the use of any animals whatsoever. So the main considerations of choosing a 3D cell culture model, because you've got lots to choose from, things to think about is, do you need a clonal growth model or do you need an aggregation model? And these are things I can discuss in the tutorial. The size of the 3D construct needed is absolutely important for some experiments. So for our mass spectrometry imaging studies, we need large constructs. For simple apoptotic studies, much smaller constructs are needed. Do you want to do monoculture, such as growing stuff like tumour cells on their own, or do you want to do co-culture? Some of our studies are moving into including tumour-associated fibroblasts and tumour-associated macrophages into our spheroid models to study in cell culture the effect of these normal cells upon tumour behaviour. You need to consider, if doing co-culture, how you're going to tell which cell is what. So you need some sort of staining, and our multiplex mass spec imaging methods that I've just shown you, I can talk about those more in the tutorial as well, but those are proving to be very, very useful for that. You need to consider how you're going to analyse what's actually occurring in the construct. Are you going to use imaging? Are you going to use ATP levels to monitor proliferation? Are you going to do immunohistochemistry? All of those are possible. Some work well, some don't work quite as well, and it's fair to say that 3D cell culture is much trickier than 2D cell culture for doing the analysis. For most of what I do, which is drug sensitivity studies, we need to know, is the drug even getting in there? Are the cells more or less sensitive in 3D cell culture, and why? And that's the sort of the primary question that I'm working on at the moment. So most of my studies are looking at either drugs or radiotherapy in 3D cell culture, and what we find is that in most situations the cells in 3D cell culture are pretty much resistant to drugs. In a few situations they are more sensitive in 3D cell culture, but usually they're completely resistant. So my work at the moment is trying to work out why cells are resistant to drugs, even though we know the drug is getting in, and also particularly radiotherapy. Why are cells in 3D cell culture much more radio resistant compared to normal cell culture? The reason being is I'm trying to create experimental models which in a way replace the need for studying cells in live organisms, particularly animal models. So that's why I do 3D cell culture, is to replace animal models in experiments, but in doing that we need to know why we're getting the results that we're getting, which are very different from standard 2D cell culture. So we'll talk about different experiments that can be done with 3D cell culture in the tutorial later this week. Okay, that's it from me.